Fertilization is the fusion of a haploid sperm and egg nuclei to form a diploid zygote cell. This event typically occurs in the oviduct of the female reproductive system, and the fertilized egg then travels through the oviduct, dividing as it goes, until it reaches the uterus, where it will implant into the endometrium and begin embryonic development. The secondary oocyte that leaves the ovary and enters the oviduct is many times larger than the sperm cell that will eventually penetrate the membrane and fertilize the cell. And what we see here is an outer rim around the secondary oocyte called the zona pellucida. This is a layer of sugar and protein that the sperm must penetrate to get through to fertilize the egg. The secondary oocyte is also surrounded by a number of these granulosa cells that came from the follicle in the ovary. So the sperm will make its way through those granulosa cells and then release acrosomal enzymes from its acrosome to digest its way through the plasma membrane of the secondary oocyte. At that point, the sperm nucleus will be ejected into the cytoplasm of the secondary oocyte. When that occurs, the secondary oocyte completes meiosis II to officially become a haploid egg. Now at that point, the egg nucleus and the sperm nucleus unite, and two sets of chromosomes come together to form the first diploid cell. So again, the sperm meets the egg in the oviduct, and that forms the diploid zygote cell. And then embryonic development begins by the rapid division of this zygote cell repeatedly as it travels down the oviduct. And these events are called cleavage, the repeated mitosis of the zygote into different stages marked by the number of cells. So there's a 2-cell stage, and then at day 2 about a 4-cell stage, until day 3 when this rapid division occurs over the course of that 24 to 36 hours, making a morula, the next stage of embryonic development, which can be up to about 100 cells. And these 100 cells are compact in one ball of cells, and the significant thing that occurs within this stage is that the morula is the structure that enters the uterus. Following the entrance to the uterus, of course, mitosis is always going on. The cells of the morula start to inject a fluid towards the center. And around day 4, the developing embryo becomes what's called a blastocyst, the hollow ball of cells. Now, inside this hollow ball of cells are a group of cells that are the stem cells. These are undifferentiated cells that will become the future organism, while the outermost cells become the extraembryonic layers. So this blastocyst is what actually implants into the endometrium. So a very significant event in embryonic development. The stem cells that become the embryo, while the remainder of the cells become the extraembryonic layers like the placenta. Approximately a week after implantation, the process of gastrulation begins. And recall that the blastocyst was a hollow ball of cells with an inner mass of stem cells that become the embryo, while the outside layers become the extraembryonic layers around the fetus. Those layers are the prechorion, which become the placenta, and the amnion, which becomes the amniotic sac surrounding the fetus. Now, the significant process that happens during gastrulation is that those stem cells that could become anything now differentiate into the 3 primary germ layers we see here. The ectoderm, shown in blue, the mesoderm, shown by these pink cells, and the endoderm here in the yellow. Now, these embryonic tissues give rise to all the future tissues of the body, and they form by the movement of cells through what's called a primitive streak that develops in many different animals. Now, the ectoderm is the tissue layer that will become the central nervous system, as well as the epidermis, that upper layer of our integument. And we'll see in the next process, neurulation, how that ectoderm gives rise to the central nervous system, the brain and the spinal cord. The mesoderm gives rise to most tissues and organs of the body. These are muscle, bone, our digestive and respiratory organs, and basically 95% of our body is derived from mesoderm tissue. The inner lining of our major organs, like our digestive and respiratory organs, comes from the endoderm, this third tissue layer. The next event in embryonic development is neurulation, and this is the formation of the neural tube, and it marks the first appearance of an organ or organ system in the embryo. Now, it all starts from the formation of what's called the notochord, which is a thin, flexible rod that extends through the organism's dorsal side. Now, it's important to remember the three-dimensional nature of the embryo at this point. So we see a two-dimensional cross-section on the screen here, but down in the lower right corner, think about that two-dimensional cross-section extending back into space, and thus that little circle that is the notochord is actually a long, thin tube, and that's going to be important for later understanding the formation of the neural tube. So what we see is the ectoderm, that outermost layer in blue there, start to fold inward. And as it begins to fold inward, and again this is induced by the presence of the notochord, it will slowly invaginate and pinch off from the external portion of the ectoderm to become its own neural tube. And of course that neural tube is what will become the central nervous system. So at the anterior or head end of the organism, that neural tube will expand to become the brain, and of course the remainder of it will be the spinal cord. Now, this step of embryonic development is also where we see the appearance of these structures called somites from the mesodermal tissue. And somites are these repeated blocks of mesodermal tissue that are the segments of the organism that will become, for instance, the ribs and the vertebrae of the thoracic cavity. And here is actually a frog neurula showing many of these structures similar to us. Even though we are very different from amphibians, we have the same similar pattern of embryonic development. And we see here the neural tube that is formed at the top, induced from the presence of the notochord just below it. And we can also see that mesodermal tissue around the sides forming the somites. Now, embryonic development then ends after about 2 months. After this, we call the developing organism a fetus, and fetal development continues. Now, most of the major organs have developed by this time, as well as eyes, some limb buds, and we also see a postanal tail. You can also see, especially in that postanal tail and between the two limb buds, the many somites, the repeating structures of mesodermal tissue that have arisen.